Hi, everyone. I'm Dr. Pedro Ramirez, Editor-in-Chief of the International Journal of Gynecological Cancer. And I wanted to welcome to today's Journal Club. I would like to first thank ESGO and IGCS for their support in our Journal Club. Before we begin, I just wanted to mention a few housekeeping notes relevant to the Zoom platform for today's Journal Club. We're recording today's discussion, and the recording will be made publicly available. Participants with the cameras turned on may appear in the video recording. So we ask that everyone please keep your microphones muted so everyone can hear the presentation. And if you're having any difficulties or questions, please send us a message in the chat so that our technical support will be able to assist. Definitely, we have time for questions and answers, and I know that our guest speaker today enjoys questions, and particularly very difficult questions. So please submit your questions to the chat. So for this month's lead article, I'm really very pleased to have Dr. Rob Coleman, friend and colleague, back in our journal club. This article is titled "Mirvetuximab in Folate Receptor Alpha High Platinum Resistant Ovarian Cancer: Final Overall Survival and Post Hoc Sequence of Therapy Subgroup Results of the Soraya Trial." Rob, thank you so much once again, obviously, for accepting our invitation. Welcome to the Journal Club. Really looking forward to your presentation. And then certainly, obviously, we want to encourage not only the questions from our fellows, but also questions from the audience. So, Rob, welcome. Great to see you again, and thank you.
Yeah, thank you so much. It's so great to be with you, and I really appreciate this opportunity. Yeah, if the questions are about like how the internal dynamics of a combustion engine, probably won't be able to answer those very well. So kind of keep them on topic. But I know Pedro can pitch in for me if that happens. So I appreciate it so much. Thanks so much, Pedro. I appreciate being here. Um, as Dr. Ramirez mentioned in this long title, we're going to talk today about the Soraya trial. One of the things I want everybody to kind of keep in mind is that there are very few kind of really neat success stories in GYN cancers. We've had a few in the last couple of years, particularly in endometrial cancer. But for ovarian cancer, many of you remember it's been kind of a desert since we've seen the institution of the PARP inhibitors in the frontline setting. We had them in the second line setting, but in most parts of the world now those have been retracted. So this particular sequence of studies on a drug that is we're very excited about, mirvetuximab soravtansine, as an antibody drug conjugate, kind of launched the re-excitement of drug development in platinum-resistant ovarian cancer. Now it's spreading into many other settings. But this is a neat story because it almost got completely derailed. And hopefully we get a chance to talk about that as we go through it. These are my disclosures, and I will be discussing, basically on-label use, which is really exciting because most of the time when you give a talk like this, it's all investigational. This is not anymore because it's now approved. So as I mentioned, platinum-resistant ovarian cancer has been obviously a very difficult situation clinically for us because once the tumors have gone through a number of lines of therapy and become resistant to platinum, they share resistance mechanisms with many other drugs. And in this space, there's been really robust clinical trial development and exploration of novel agents, which have really resulted in some nice signals, but almost none of these have actually been confirmed. Outside of our kind of traditional chemotherapy single agents and in combination with bevacizumab for appropriate patients, our chemotherapy alone response rates have been really in the single digits to low double digits. This basically, provided, the background for why is there so much robust development from a preclinical situation to try to identify, targets that would be of relevance. And in ovarian cancer, it was discovered many years ago that ovarian cancer in general expresses a high degree of folate receptor alpha, and this has been used as a target for other drugs in development over the years. Actually, the relationship between folate metabolism and actual tumor biology started back with the recognition that methotrexate could be active. And so you can imagine how many years ago, how many decades ago that was. But the antigen substrate for folate receptor was also in development with other drugs such as ventafilide. And those, again, for the most part, most of these drugs have not progressed. So the idea of coming up with an antibody that targeted folate receptor alpha combined with a drug which was felt to be very potent and directive to ovarian cancer biology, such as the maytansine analogs, in this case DM4, were put together in a molecule that was connected by a cleavable linker. The idea here was that we could selectively deliver a potent tubulin targeting agent directly into the tumor because it would target those particular cells that express folate receptor alpha. So SORAYA was designed as part of a long story of trying to develop this drug in ovarian cancer when it was realized that expression of folate receptor alpha could be measured by different assays and to what degrees were going to be important. So when this program was rebooted, we came up with the idea of doing a phase 2 and a phase 3 confirmatory trial, which is MIRASOL, to try to demonstrate that indeed this drug did have efficacy and could win. So SORAYA was designed as a phase 2. It was purposely designed by sample size. I'll discuss that in a second. And because of its efficacy with respect to response, to duration response, and safety, it was achieved a accelerated approval as a result of this trial, and then received its regular approval as a result of the confirmatory trial known as MIRASOL. So this particular trial, as I mentioned, really grew out of a re-evaluation of the entire mirvetuximab program. Many of you remembered that Forward 1 was a randomized phase 3 trial of mirvetuximab versus chemotherapy, which did not meet its primary endpoint. And there's lots of reasons for why it didn't. I'd love to discuss that as well. But it almost completely crashed. And I remember sitting in an airport lounge at JFK Airport with the company saying, "Oh my God, what are we going to do now?" And we designed SORAYA. We designed this with the intent of providing a sample size that was large enough to provide precision around the point estimate of objective response. We wanted to have robust assessment of the safety of the drug, knowing that ocular toxicity was an issue. We wanted to demonstrate that patients who responded to the drug had a long duration of response. And we wanted the trial to identify patients whose tumors were most likely to respond. And that was the change to the PS2 evaluation of folate receptor alpha expression. In this case, that we had to have at least 75% of viable tumor cells expressing at least 2+ staining by IHC. And this turned out to be the Ventana FOLR1 assay, which is now available to many of you. The one other component of this that we felt was very important, and it was a strategic decision, was that we required that all patients enrolled in the trial have been exposed to bevacizumab. The reason I say that is that in this particular single-arm open-label trial, we needed to show that patients who've been previously exposed to bevacizumab would not necessarily be eligible for a chemotherapy combination. So we wanted a level playing ground that allowed us to compare the results of this trial as a single agent against the expected historical response to chemotherapy alone and not with bevacizumab. And because PARP inhibitors were being used more prolifically, we allowed that in the trial to provide a more diverse population. Now, on the right-hand side, you'll see what the treatment schedule is. This is the standard dose that we're continuing to do right now. I will highlight, if there's any interest to the people who are listening in, that this adjusted ideal body weight of 6 milligrams per kilogram was a dose modification, but did not go through the formal dose optimization program that is now required by FDA. And we could talk about that as part of the post-marketing commitment for the drug. 110 patients were actually planned to be recruited. We wanted over 100 because of the, as I mentioned, the confidence intervals. We wanted to be very precise, and we wanted the power to detect a 24% response rate. These are the patients that went in the trial. There were 106 ultimately enrolled, 105 were evaluable for the efficacy endpoint, 106 for safety. And as you can look down this list, you can see why I was so deliberate in my comments about how we chose the patient population. Most of these patients had three prior lines of therapy. All of these patients were exposed to bevacizumab in the past, either as part of their platinum sensitive frontline or platinum resistant recurrence regimen. Half of these patients have been exposed to PARP, and nearly all were exposed to taxanes. So this is important because ZM4 is a tubulin poison. So these would be administered to patients who have been previously treated with the other tubulin poison. You can see that 63% of these patients received mirvetuximab at the first point that they became platinum resistant. And we define that, as you can see down at the bottom, as patients who had not progressed well on frontline platinum, that had failed within 6 months of their most recent platinum exposure. And as I mentioned, since bevacizumab could be given during at any point in time, 90% of these patients received it as part of their platinum-sensitive ovarian cancer regimen. And you can see again with their platinum-free interval and the primary platinum-free interval that many of these patients were primarily platinum-resistant to begin with, over almost 60%. And of those that had a platinum-free interval from the most indexed treatment, a third of these patients were within 3 months. So a fairly significant platinum-resistant population. Again, the point of this, as I want to emphasize it, is that we wanted this to be aligned with what you would have considered as the other therapy if you didn't have access to this study. What would you treat your patient with if you had a patient with these characteristics? That was the point. Now, the paper itself, we talked about overall survival. I want to get to that, but I do want to reiterate what was really important for the development of this agent. The investigator assessed objective response rate for the trial was 32.4%. Again, because of 105 patients, the lower limit of confidence was at 24%, which was actually where we were targeting our expected response rate, because we wanted to show that we could beat a 12% response rate from single agent chemotherapy. So this was very exciting to see. It was one of the reasons why it was granted accelerated approval. On the right-hand side, you can see that the patients who did respond, of those 34, they responded at a median of 6.9 months. Again, 95% confidence interval between 5.6 and 9.7 months. Again, this is far greater than we would have expected. When we broke this down by when patients received mirvetuximab in this resistant setting, if this was their first treatment, you can see what the number is. The response rate was at 35%, and if it wasn't, then it was a little lower at 28%. And then when we also looked at this with respect to when bevacizumab was used, and since this was more often given during platinum sensitive, you can see the response rate immediately post or following bev exposure was 34%, and lower for the platinum resistant cohort when bev was given with platinum resistant ovarian cancer. And this probably shouldn't surprise you, because patients that are receiving platinum resistant ovarian cancer with bev would have received it as their second or third line therapy, right? Or second line therapy or first line therapy in platinum resistant ovarian cancer. So this would have been a patient that was actually more resistant, so to speak, than the platinum sensitive patients. Waterfall plot demonstrating the responses over the course of the trial. These are the best responses in the patients that we had data for that. And then getting to the overall survival. So we initially reported overall survival. It was immature. You can see now that with most of the censoring events are occurring at 18 months and beyond. And you can see the curve is very steady with the median overall survival of 15 months. Again, this was something that we were very excited to see, because in this particular setting, in patients who have also progressed on bevacizumab containing compounds, we were expecting this to be somewhere between 10 and 12 months. So to see a median 15 months was very exciting. Really exciting is that at 24 months, you can still see that about a third of the patients still were alive, which is actually quite remarkable for a PROC trial. We broke this down also by number of prior lines. And again, this shouldn't be a surprise to anybody. The more lines of therapy, the lower the expected survivorship. We know that just in terms of expectations, we lose about 20% of our patients per line of therapy, especially in platinum resistant setting. So, but you could see there was this change in difference. And these are not independent factors. We'll talk about that, I hope, in the discussion. that patients that have received 1 or 2 lines of therapy versus those who received 3 lines of therapy also have other characteristics which make them have poorer prognosis. So something just to keep in mind. Something that, again, we were happy to see with longer follow-up, this added to the global database of patients who have been ever exposed to mirvetuximab. So this was obviously accumulated before Mirasol was put into the picture. But this was combined with all of the, the Forward 1, Forward 2, and other trials that were used that used mirvetuximab. Fortunately, we didn't see anything new. What I will say is that, the ocular toxicities, despite the fact that we've done better education and we were obviously very in tune with that adverse event for this trial, the rates were basically the same. The overall rates of being around overall ocular events being about 40 to 50% of our patients, most of it being, or about half being grade 1 and grade 2. Fortunately, these were all reversible, but the other toxicities that we might have expected with a very toxic chemotherapy, such as hematologic toxicity, neutropenia, vomiting, other GI side effects, were actually quite low. And we're very excited to see that the selectivity of the ADC was actually borne out with this regimen. To dig in a little bit or double click into the ocular adverse events, because I mentioned about half the patients had blurred vision. This is probably the most common of the side effects. In fact, the one patient who had the grade 4 was really about a diminution in her visual acuity. It went from I don't remember what her baseline is, but it went to 20 over 200, which was considered a grade 4. I had several patients who've had this, event and not tell me about it. So you almost have to ask about it, but it was definitely an issue that we saw. It was predictable. It came on usually on the first or second week of the second cycle. And all of these essentially resolved very manageable. Fortunately, the number of patients that had to discontinue on the study overall was only about 9%, but to see them discontinue with respect to ocular events, it was just a single patient. So again, we've gotten quite good at this, and this was very reassuring. So in conclusion, this drug, as a single agent, demonstrated very robust efficacy benchmarks as assessed by the investigator for both objective response, duration of response, and impressive overall survival in patients who had had prior treatment, many of whom have had prior treatment in the platinum-resistant setting. The numbers are very relevant given the patient population, and this was added, as I said, to the collective database on mirvetuximab, which showed a very consistent, reproducible adverse event profile that allowed us to keep patients on therapy where they could benefit the most. So this trial, as I mentioned, did lead to an accelerated approval, so it became available in the United States. So it's actually quite exciting that we were able to get Mirasol completed. One thing I will say parenthetically in the development of this drug was that the FDA actually looked at the data in the Mirasol trial in the experimental arm to confirm that what they saw here was not a fluke. That doesn't happen very often, but it was a posture that the FDA took because they didn't believe it, to be honest with you. And so it was very important to see that that was confirmed, and ultimately this became. So really, I want to, as always, thank the patients and the families that participated in the SORAY trial. It really was a landmark for us and provided the first step to what is now an explosion of ADC development in GYN cancers, as you probably well know. And this first drug, obviously now, is material to the development of these other ADCs because we have to exclude patients who are actually eligible for this drug if we want to assess future ADCs in the space. I also want to give a thanks to Pedro, Dr. Ramirez, and the journal for accepting this paper for publication. I do think this is really an important trial, and the visibility that was granted by the journal has really helped us quite a bit. So thank you so much, Pedro. And I also want to thank all the multiple sites that participated in the trial. And with that, I'll stop and happy to take questions.
Excellent, Rob. Thank you so much for that. Very, very thorough presentation. As always, obviously, you're very clear and articulate the results of these studies very well. So we'll start with the questions from the fellows. And again, also want to encourage the audience to participate and send in questions as well. So the first fellow that is going to be asking her question is Osnat El-Shavi from Israel.
Hi, and I will join congratulations and thank you, Dr. Coleman, to you and your co-authors for this important contribution.
Thank you.
My question is that given the patients in the trial were required to have their archival tissue tested for folate receptor alpha expression, and considering the decreasing response to mirvetuximab with each additional line of treatment and the potential development of drug resistance, do you think that folate receptor alpha expression may diminish over the course of the disease? And how might this affect the interpretation of the results?
Yeah, great. Very thoughtful question. And brings up, many different points. Let me just start with the first about the relationship between number of lines of therapy and the results that we saw from the overall response rate. So as I mentioned in the presentation, it really shouldn't be surprising that patients, for instance, who are getting this drug as their third line of therapy, wouldn't have a lower response to treatment. That's been observed with every drug that we've studied. In fact, we actually also can see it even in the platinum sensitive setting. So drugs that share common modalities of drug resistance typically will affect the subsequent lines of therapy. So that shouldn't surprise you. I think if anything, the fact that we saw almost an 18% response rate in patients with third line is actually a very strong tribute to the efficacy of the drug, because in third line patients, we expect to see response rates around 5 to 8%. So again, this is higher than expected. The second question really has to do with the robustness of antigen presentation. So we don't really know sequentially what happens to the folate receptor on tumor specimens themselves. There is some data to suggest that because it's so prolific on the expression on specific cancer cells, that there may be a heterogeneity that gets induced when patients are treated with a folate receptor alpha targeted ADC over time because of the selectivity of targeting. But we don't know that just without treatment with an ADC that's targeting folate receptor alpha, whether or not that actual impacts the overall expression of folate receptor alpha for multiple lines of therapy. So we don't know if it's our interrogation and use of a drug that hits the target itself that potentially changes expression, or if it's just the expression itself with tumors that recur and have previously been treated with other lines of therapy. So it's a bit of an unknown there. But I do think that, to answer your question more specifically, the reduction in objective response rate to me is probably a greater reflection of the types of patients who had third line therapy, what they responded to in the previous, have they just progressed off a platinum resistant regimen and would have a lower likelihood to respond to the next line of therapy.
Excellent. Thank you, Osnat. And Rob, I wanted to just jump in also with a question. It's my understanding that when you look at archival tissue versus tissue that is obtained at the time of recurrence, there's often about like a 70 to 80% concordance with archive versus tissue. But I believe I read that if you were originally folate receptor negative, that there could be up to like 40% of patients that then at the time of recurrence are folate receptor positive. Have you had any experience with that?
We have. Obviously, there's a greater robustness with retesting tumor tissue. For the most part, it is a lower number. That I think 40% is probably an upper range, mostly because folate receptor alpha is kind of ubiquitously expressed in across almost every, most of the cells that cancer cells themselves. So to have a truly negative might be an assay issue, and that is something that we see, is that the assays with older tissues can sometimes lead to disparate or inconclusive results. So yeah, it would have to be really well paired and hopefully in the same area, because actually tumor heterogeneity in and of itself can actually lead to some of those discrepancies. However, because the drug is so important, patients who have a negative, quote, original assessment, I probably would repeat on a tumor biopsy just to make sure.
Okay, great. So the next question is from Susan Dababo in Italy.
Hello, hi, and thank you, Dr. Coleman, for the great presentation. And I would like to ask you, with the completion of the MIRASOL trial, how do the efficacy and safety results compare to those observed in the SORAYA trial? And what key insights have been gained from the MIRASOL and SORAYA trial that might influence the clinical application of mirvetuximab soravtansine? And how do these findings impact future research direction and treatment protocols for ovarian cancers? Thank you.
Yeah, great. Yeah, several great questions there. So let me start with the first one. As I mentioned, that the SORAYA trial got its accelerated approval based on the results of this trial. But while this trial was under consideration by the FDA, the results of the MIRASOL trial in the experimental arm were being looked at. So if you think about that, you had one trial that was done, had results published, and then you had another decision by the FDA that said, wait a minute, we're going to wait until the other trial actually gives us enough data with enough maturity to confirm what we saw in the first trial. So when it was granted accelerated approval after that sequence of events, we already knew that MIRASOL was going to be very reflective of the SORAYA data. If they were discrepant, the FDA would not have granted the accelerated approval. Okay, so in general, although the numbers are not exactly the same, X response rate is a little higher, it just goes to show that there were some differences in the patient population. The most notable difference was that we did not require bevacizumab to be administered in all the patients enrolling on MIRASOL. So they may have had less prior exposure or less intense therapy prior to participation. Nevertheless, the data that we saw from overall survival, the data we saw from progression-free survival, the duration of response, these numbers are generally within the same ballpark. Noting that there are just some slight differences in the patient populations going in, including PARP exposure, et cetera. So those were the main kind of differences. We knew they were going to be aligned, and so if anything, that should just bolster confidence now that we have 300 patients treated essentially with a single arm with the drug. The next question is that really gets back to how this influences future research. And I think I mentioned this already, that one, it basically resurrected the concept of ADCs. And as you know, right now, I think there are probably 7 different ADCs in ovarian cancer development that are now, instead of targeting just folate receptor alpha, which there are 4 drugs that are targeting folate receptor alpha as ADCs. We also have TROP2, CDH6, Claudin-6, and HER2. All of those are now ADCs under active in phase 2 to 3 investigation in ovarian cancer. So this had profound effect on bringing that drug strategy back to the diseases that we treat. The other important impact of this drug and this study platform was that now that this drug is approved, it's considered in the United States, it's considered available therapy. So you can't do a physician's choice chemotherapy trial unless you include patients who have FR alpha high expression and get mirvetuximab, or you have to add mirvetuximab to the control arm. So if you think about that, we have a response rate from both out of SORAYA and out of MIRASOL that's about 2 to 3 times higher than standard chemotherapy. So if you're evaluating another drug in this setting, the bar is much higher to reach. So our clinical trial development strategies now are to exclude patients who, or require them to have been previously treated with mirvetuximab if they're FR receptor alpha high.
Great, thank you so much, Rob. And next is Matteo Pavoni from Italy.
Hi, thank you for sharing your results, and congratulations for your study. My question is, what additional studies are needed on MIRV in patients with platinum-resistant ovarian cancer? And additionally, where there are any biomarkers identified that might predict response or resistance to MIRV therapy, which could inform patient selections in future trials?
Yeah, man, another fantastic question. Questions. So let me start with the first. Right now, if you think about this as just being chemotherapy, and let's say you have a new chemotherapy that you've just shown has efficacy, what would be your natural next step? So the first natural next step would be whether or not there was a combination that would be helpful. So the combination strategy evolved around 3 different pathways. One was to use it in combination with bevacizumab, obviously, because that's what we have used with the Aurelia trial and all of our chemotherapy agents, and was actually excluded in the trial, right? A requirement for prior bev exposure. So that was one pathway. The second was to try to combine it with another chemotherapy. And obviously, if you were thinking about development of this agent, you might think of it as a replacement for paclitaxel. So again, if you're thinking about how you would develop this drug, what would you want to combine it with to replace, if you were going to replace it for paclitaxel? And obviously, then the biggest drug that we would want to, align it with would be platinum. So it aligned down the platinum angle. And then the third angle, because at the time that this drug first came onto the scene, there was a lot of interest in whether or not chemotherapy could change a kind of a cold tumor microenvironment for immunotherapy into a hot environment. And so it evolved also along the use of concomitant therapy with an immune checkpoint inhibitor. So those were the 3 development lines that went, that started this story. And right now, in the landscape, we really haven't continued to pursue the immunotherapy, but Bev was very important. And so MIRV and Bev now has been instituted into, as you know, the Gloriosa trial as a maintenance therapy, as a combination for maintenance therapy in platinum-sensitive ovarian cancer. We also have efficacy data with platinum and with bevacizumab in the recurrent platinum-sensitive environment. And so those are actively ongoing. And then I think it's now in the public domain that there's interest in developing 2 trials in the frontline setting. So again, trying to figure out a way that if we can replace paclitaxel and figure out a way that we can use it as a maintenance therapy, which is not easy. Remember, this is chemotherapy. It's not like a biological agent like bevacizumab or hormonal therapy like letrozole that we can use, endlessly. So toxicities will be an issue that we will run into it that we have to address in these other longer exposures. So those are kind of the 3 kind of primary development lines. The fourth one has to do with the biomarker. So you asked, like, what are other predictors for mirvetuximab? It wasn't like a light switch was turned off if they were less than 75. What we found was that the response rate were highest in tumors that had 75% expression of 2+ or more. And that's why we focused on that cutoff to do this development strategy. We didn't want to lose again because we lost once and we didn't want to lose again. So we chose the patients most likely to benefit, and we use those as our eligibility concern. But because the response rate is actually not too bad in the 50%, there's also been an interest in wanting to look at other tumor expression profiles for which these drugs might have efficacy. And so that's also being studied with this. So those are the 3. So combinations, lower biomarker, and then a different line of setting.
Excellent. So Rob, we have next is Ana Luz Arraga from Barcelona.
Hello, Dr. Coleman. Congratulations for this excellent paper and presentation. So my question is a little bit related with what you already said. So that as the objective response rate was higher in patients who received mirvetuximab as the first treatment in platinum resistance versus those who initially received another treatment, do you think it is justified moving mirvetuximab into the platinum-sensitive setting, as in the Gloriosa trial, or even considering it for the first-line treatment? Thank you.
Yeah, thanks for this question. It's actually really timely with respect to development strategy. I know I keep going back to that. But I think it's helpful for you guys to understand how these drugs actually get developed so that you can use them in the clinic. And hopefully, if you have opportunity to participate in clinical trials, you can follow, try to help to enroll patients into trials. There was a trial that was recently put before the FDA that was looking at in non-small cell lung cancer, the combination of a drug, chemotherapy, plus a new drug, and it was given in the neoadjuvant setting. And then the patients then had surgery, and then they continued on the new drug. And the concept was that they said to the FDA, they said, hey, listen, we want this drug approved in this setting because it works neoadjuvantly and as adjuvant. And the FDA said, okay, yeah, we see what you're doing here, but we want to have somebody else look at this data and confirm whether or not it's important to sort out the relative contribution of when the therapy was given. And they voted 14-0 to do that. So it was a surprise to us that in a couple of days ago that the FDA approved this drug combination as a neoadjuvant adjuvant combination without requiring them to go back and say, you only need it during adjuvant, or you only need it during maintenance, or that you need it in both. So to do that kind of a trial requires 4 arms. It's kind of a 2x2 design. So the reason I say that is that in the trials that we're designing right now that are trying to replace paclitaxel, the paclitaxel carboplatin combination, or paclitaxel carboplatin bevacizumab combination, is being given in the front line before the maintenance. And so 2 questions will be asked, is that if the bevacizumab, mirvetuximab maintenance strategy is efficacious, we're going to need to also prove that it is beneficial before they get the maintenance. And so we don't know exactly whether or not this is going to be a problem for us, but the strategy here is to figure out a way to move this all the way into the front line, to get rid of Taxol, and to see if we can give this as a induction treatment and as a maintenance treatment like we are in Gloriosa. So lots of considerations there. I know it's kind of complicated if you can't see it on paper, but the idea here is that we are trying to prove not only the contribution of effect of the new drug, MIRV, but we also want to show that you have to give it in both settings. And that's what's ongoing right now.
Excellent. And next question is from Melissa. Melissa, I don't know if you want to, because I think that Rob already sort of addressed this question, or if you wanted to ask him an extended or a different question.
Yeah, he partly, and thank you, Dr. Coleman, for this great presentation. As Pedro mentioned, he already answered my question, so maybe we can skip to the next question if you want.
All right.
That's okay. I'm happy to address if you have any curiosities. First of all, where are you? What part of the world are you in?
In Istanbul, Turkey.
Excellent. Yeah, or if you think of something, I'm happy to address it or put it in the chat. I'd be happy to address it.
Excellent. Okay. So we'll move forward to Austria. Natalia.
Hello, and thank you very much. Thank you for sharing your knowledge with us, and congratulations on your work. My question is, what do you think about mirvetuximab in neoadjuvant treatment? Is there potential there? And for today, do you think that this therapy should be the first choice for treatment in the platinum-resistant patients in clinical practice?
So say this last one, you kind of cut out for a second. The last part of your question?
If you think that this therapy today should be the first choice for treatment in the platinum-resistant patients, like in clinical practice, you have a patient today, are you doing this?
Yes, first of all, I am. And I think that the major question is, would you give this drug as a single agent, or would you give weekly paclitaxel and bevacizumab? Because they have about the same overall objective response rate. And I think, in patients that have the expression of the folate receptor alpha, I would be inclined to use it there. And I would actually, again, be looking for a clinical trial where I could give it as a combination. We do have safety for the combination. But I like it because the adverse event profile is a little bit nicer for us, and especially with respect to hair loss and myelosuppression and things like that. So I think the neoadjuvant question, as I mentioned before, right now there's interest in wanting to do this in the frontline setting right now. So that's already kind of a part of this process. But but you can make a case for this even in the recurrent setting too, especially if you're considering secondary cytoreduction, which we're not going to talk about, argue about today. But if that was part of your strategy, that would be another potential opportunity. Mm-hmm.
Great. And then Rob, Natalia is going to follow up with another question. This
All right.
is that was sent from Rosa Montero. Natalia?
Yeah, I actually already asked both. So
okay, perfect.
The clinical practice question was originally from Rosa, stolen from Rosa.
Okay, perfect.
Natalia, let me ask you: do you what's the status of mirvetuximab's availability in Turkey?
No, Natalia is in Austria.
Oh, Austria. Oh, okay, Austria. Okay.
Natalia may know what is the status in Turkey.
Yeah, what is it? Yeah, what is it, Natalia? What is it in Austria?
I don't know what it is in Turkey, but in Austria, like you have to ask for it, and it takes a lot of work to really get it. So it's a lot of paperwork that we have to do to get it.
Okay.
Excellent.
Okay. Yeah.
So Rob, we just have a few more minutes, so some kind of like rapid fire questions from the audience. So first one is, do you have a sense that the response rate in platinum-resistant ovarian cancer is higher, different than in platinum-sensitive ovarian cancer?
In general, yes.
Yeah.
In general, okay. But it's also, again, it's related to the platinum, whether or not platinum is used or not. Platinum versus non-platinum and platinum sensitive, platinum wins. Platinum versus non-platinum and platinum resistant, sometimes platinum wins. But this is actually, if you look at mirvetuximab, this is actually probably better than we would expect with platinum.
Okay. Next question. What are you giving after progressing on mirvetuximab?
Oh my gosh. Yeah. Clinical trial. So one of the things I would say is that if they haven't received, weekly paclitaxel, it's kind of my go-to. I like gemcitabine and cisplatin as a doublet. That's one of my go-tos. If Bev can be used again, that's another potential go-to. But I do think that what we're trying to do now is to get a broader, sequencing array for patients, as well as IHC, to see whether or not these patients could be eligible for drugs that are also available, such as trastuzumab deruxtecan or others.
Okay. Next question. I'll kind of say it slowly because it's kind of a detail. In mirvetuximab, antibody drug conjugate acts against the folate alpha receptor. It's the DM4 that kills the cell. If MIRV also has anti-neoplastic activity, then what kind of interaction is between them? Is it additive or synergistic?
Yeah, good question. So yes. Right now, the predominant mechanism of action for mirvetuximab soravtansine. So it's those two components. MIRV is the antibody that's targeting, which, as far as we can tell right now, because of our experience with other folate receptor alpha targeted antibodies, has very little efficacy. So if you give just the antibody, this is similar to what we saw with farletuzumab, which was just a folate receptor antibody. We don't really see much in the way of efficacy. We've done 3 or 4 now phase 3 trials that shows that that's not the case. So the DM4 is the thing that's causing the cell kill. Now, I'll say that for this drug, but what's happening in the ADC world is that we're getting very smart at developing new antibody or antibody-like drug conjugates, where the antibody itself can induce an immunological response that's separate from the release of the drug itself, the DM4 or whatever the other drugs that they're connecting to the antibody. So this space is going to continue to evolve because it provides tumor selectivity and delivery of assets. The other thing I'll just mention really quickly that's kind of related to this question is that we have what's called bispecific antibodies. that can also be loaded with drug conjugates. These drugs, there was actually one approved today in non-small cell lung cancer, again, that targets EGFR and c-MET, so it has a double hit. These are also being developed in ovarian cancer for multiple targets such as VEGF and EGFR, or VEGF and some other components. So these are new developments in the ADC world. But for mirvetuximab, the money part of this is the DM4.
Excellent. Well, Rob, thank you so much. We're coming to the conclusion of the Journal Club, and I really, truly appreciate your input. Always learn so much, of course, from you and having these discussions. Truly want to thank all of the fellows for your questions and for your discussion during the Journal Club. I also want to remind everybody that the recording of today's session will be available in the IJGC, IGCS, and ESGO websites. Also like to remind you that the September Journal Club is coming up. It's with Kika Chacon from Spain, and the title is going to be the Seneca Study, Staging Endometrial Cancer Based on Molecular Classification. So thank you so much once again. Thank you, Rob. Really always a pleasure.
Same. Thank you so much for having me. It's great to see all these young faces and inquisitive minds. Love it.
All right. Take care,
All right,
everybody.
buddy. Take care. You too.
Bye.
Bye-bye.